4-part webinar series on microplastics. Today's webinar focuses on how microplastics affect the species and environments that they interact with. And previously, we learned about how microplastics flow through the mid-Atlantic waterways, how they move through the marine food chain, and now what impacts microplastics have on those species and habitats. I'd also like to extend a big thank you to the members of Marco's Marine Debris Work Group and our microplastic fellow, Joseph Barnes, who's here today as well, who collectively put together a lot of time and thought into creating this series. The work group is made up of state and federal agencies, as well as NGO partners that are working together toward a shared goal of bringing together regional stakeholders to collectively identify, understand, prevent, and mitigate the impacts of marine debris. Zooming in a little bit to today's webinar, we're going to highlight the work and research that's helping us understand what impacts microplastics are having on the physiology and toxicology of aquatic species and sediment ecosystems. And this is both through field and lab-based studies that happen in our Mid-Atlantic region. We will then have a facilitated discussion and question and answer session where we encourage you to ask the expert presenters questions about their work and also explore how regional partnerships can help advance efforts to prevent and reduce microplastics in our Mid-Atlantic region. Now, before I pass it over to our moderator for today, just a few quick notes on housekeeping. We are recording this webinar, as you might have just heard, and we'll be posting it on the Marco website. As always, please keep yourselves muted during the webinar. It's a standard practice that we're all very used to, but it never hurts to double check. As mentioned previously, there will be an opportunity for Q&A following the facilitated discussion portion of the webinar. So please put your questions in the chat box in the bottom tab of the Zoom window, and you can do that at any point throughout today's webinar. This webinar is all about offering collaborative opportunities, so feel free to also put your name, organization, and where you're tuning in from into the chat so we can all make some connections and see where our partners and friends are. With that, I'm very pleased to introduce to you today our moderator for today's webinar, Kelly Summers. Kelly is the Senior Watershed Coordinator at the U.S. Environmental Protection Agency Region 3 Mid-Atlantic Office. She leads the EPA's Regional Trash-Free Waters and Microplastics Program, where she works with partners and stakeholders to address the emerging issues trash pollution and microplastics may be having on watersheds and their resources. Additionally, she serves as the acting chair of the Chesapeake Bay's Plastic Pollution Action Team, making for a wonderful moderator for this webinar. And so with that, I'd like to thank you, Kelly, and pass it over to you.
Thank you, Will. Thumbs up if you can hear me. Yes, awesome. And thanks to everyone online for attending. We have 3 excellent speakers today who will discuss the impact of microplastics at different scales and in different environments. I'm excited to introduce our speakers now. They will each have about 10 minutes for their respective topics, and then we will have a facilitated discussion, as Will mentioned, followed by questions from the audience from the chat box. So I highly encourage you all to put questions in the chat, and we'll be fielding them as we go through. Our first speaker is Dr. Julia Fontana. Julia is a postdoctoral scholar at Oregon State University studying zooplankton ecology with a focus on how anthropogenic stressors impact zooplankton physiology. Her research spans both estuarine and polar ecosystems. She earned her PhD from the University of Delaware, where she investigated loading of microplastics through terrestrial watershed modeling, the aggregation of microplastics in estuarine systems, and their ingestion and physiological impacts on estuarine organisms in the Delaware Bay. That is a mouthful. Julia's current work focuses on energy allocation and reproductive development in Antarctic krill, exploring how krill prioritize energy reserves under varying food conditions. So Julia, you're up.
All right, thank you for that. Sorry for the mouthful of my intro. All right, let's see. Is everyone seeing this okay? Cool. All right. Well, thank you so much for inviting me. I'll be sharing a little bit, like they said, about my dissertation that was done at University of Delaware. And so part of it was looking at how microplastics and temperature may impact estuarine mysid species, Neomysis americana. Oh. Can I go forward? Oh, I'm drawing lines. Great. What's happening? Oh, hang on. Sorry. We thought we got all the kinks out. And now I lost my cursor. Here we go. There we go. So when we came up with this idea, we came up with we were thinking of this idea of compound stress. And so plastics and temperature both act as stressors on the environment. We know that they can suppress the metabolism of organisms, and we know that in aerobic organisms especially, they end up requiring more oxygen and energy to keep steady state at higher temperatures and plastic concentrations. And unfortunately, we all pretty much know that sea surface temperatures and plastic production are continuing to increase and have been increasing since about the 60s. And in that vein, organisms, or anaerobic, aerobic organisms have what's called a thermal optimum. And so as temperature increases, we're going to have this window in which our processes, our metabolic processes, are performing at its best. And so there's a temperature in which that we're reaching max capacity of oxygen intake and energy output and going to all these things that we need in our body, such as reproduction, growth, maintenance, and stuff like that. But under stressed conditions, we might see this optimum decrease and this thermal window close in. And so we'll have less of this temperature that we're able to sustain, as well as an aerobic performance at a temperature in which might have been our optimum, may be lower and reduced. And so we took this into the field. So most studies end up testing microplastic exposure or impacts in the lab and dose their animals with plastic that way. However, we wanted to see kind of the opposite of that. We wanted to see what it's like in the field at that time and what the plastic is like there. Is it impacting mysids now? And so that brings us to our question of, are current microplastic levels affecting mysid physiology now? And so the mysids we used are Neomyces americana. This might be a pretty common species to most of you. They're extremely abundant estuarine species and coastal species. They're opportunistic omnivorous feeders, so they'll feed on detritus, phytoplankton, anything they can get their mouths on. And they're preyed upon by economically important species such as striped bass and weak fish. So they're pretty important to this plastic question of, if they're going to be impacted, they're going to impact these larger trophic levels, or these higher trophic levels as well. So when we came up with this experiment, we've had this previous data that goes back to 2017 looking at the Delaware Bay and how much plastic is in it and where it ends up. And so this is what this is here. And I'll just pop this there if you want some extra reading. But here we're looking at a gradient of temperature, sorry, not temperature, plastic concentration in the Delaware Bay. And so you can see this darker red orange is going to be higher concentrations of plastic versus that yellow orange color is going to be lower concentrations of plastic. And we can see this breakout here in the upper Delaware Bay, Delaware River area, kind of north of Dover, north of Bowers Beach, between Wilmington and Bowers Beach here, where we're seeing high concentrations of plastic pretty consistently. Versus down in Lewis or towards the New Jersey coast, we're seeing far less plastic in these regions. And so taking that into consideration, we sampled from these two regions of plastic where there's high plastic and low plastic concentrations. And so Woodland Beach is the high plastic concentration we sampled at versus the mouth of the Broadkill River at Lewis Beach. And we sampled this in the winter of 2024. And we also sampled microplastics. So our actual concentrations of microplastic in the water at that time was 19 pieces per cubic meter at the high plastic location, which is a relatively high concentration of plastic, versus 0.03 pieces per cubic meter at the mouth of the Broadkill River in Lewis. And so we definitely have these regimes of high plastic versus low plastic in the lower bay. So we sampled mysids from those two regions, and we measured their metabolism between 2 and 10 degrees C. And we measured respiration, nitrogen excretion, and the atomic ratio of both respiration and nitrogen. And that was to get at this thermal performance curve, trying to see if as temperature increases, what does their metabolic do? So with respiration, If you're not familiar, respiration is just oxygen consumption. So you can think of if you're working out, if it's really hot outside, you're going to be breathing in more, you're going to be respiring more, consuming that oxygen to create energy in the form of ATP in your body. And what it looks like in practice, so these mysids were tested between 2 and 10, like I said. So here on the x-axis, we have temperature going from 2 degrees to 10 degrees and respiration rate on the y. These are typical respiration rates. They're not too outlandish. However, you can tell that from 2 to 8 degrees, they're relatively low, and 10 degrees, we start to see a peak in respiration. And it's winter, so we are expecting a repression of their metabolism in the first place. So what we think is happening on this thermal performance curve is that we're approaching at 10 degrees, we're starting to approach our thermal optimum. And I'm sorry, that was the low plastic location. This is the high plastic location, which looks very similar. However, at 10 degrees especially, we're at a lower respiration rate than the lower bay. And that may be due to a repression in their metabolism up in the bay as they are exposed to higher plastic and not used to these warmer waters. And so as the temperature increases, we're seeing either a repression of metabolism or they are lower down on that thermal optimum um, curve than the low bay, low plastic regime. And then to double check if the thermals, if these animals were thermally sensitive, we wanted to look at Q10s. And so a Q10 is just a respiratory quotient that can indicate thermal sensitivity. And so you're going to expect a twofold increase per 10-degree change in a healthy organism. So at 0 degrees, if your respiration rate was 1, at 10 degrees, you're going to expect a respiration rate of 2. But when we tested our Q10s, Woodland Beach, our high plastic location, came out with a 11.6, and our low plastic location came out with an 8.1, which are both wildly larger than a Q10 of 2, which indicates a highly indicative of thermal sensitivity for both of these regions, not just Woodland Beach. But then we wanted to get to the nitty-gritty of looking at the atomic ratio of oxygen to nitrogen. And so this is just the moles of oxygen consumed versus the moles of ammonia excreted or nitrogen excreted as ammonia. And this is to determine the primary catabolic substrate, all big fancy words to see what were they eating, what were they metabolizing. So carbs, most people are familiar, they're going to be your sugars, your starches, those are going to be your high use energies. And we'll see if an O to N greater than 50 if they're metabolizing carbs. If they're metabolizing lipids, it's typically an O to N of 20 to 50. That's going to be your fats and your oils. That's going to be your long-term energy use. Versus proteins, that's going to be an O to N of less than 16. You're low on your energy. You're using up your internal stores of muscle tissue to keep you going and to keep your body functioning. So our results here, so this is the low bay, low plastic location. We're seeing relatively low O to N ratios here. So we're seeing around 0 or like 5 to about 20. And that is symbolic of protein catabolism. However, at 10 degrees, we are seeing this lipid catabolism come into play. And so that's at the low bay location. We typically would want to see a 20 or higher value to indicate a healthier organism as you're catabolizing all of your lipid, proteins, and carbohydrates at the same time. Um, However, when we look at the high bay location, we're seeing this repression of our O to Ns. And so, especially at that 10-degree location, it's more indicative of protein catabolism, meaning that they're eating their own muscles to stay stable at this temperature. So all that to say that this high bay location is more likely stressed and more likely showing some signs of uh, metabolic stress than the lower bay low plastic location. So to tie us together here, the plastic exposure may not be impacting mysid physiology as that's not exactly what we tested here. However, that's not to say that cannot happen in the future. And it is pretty clear that these uh, mysids from higher plastic locations are catabolizing protein when warmed, meaning they're probably towards that unhealthier scale. Versus temperature is probably what is actually at play here. So we know that these warmer winter temperatures could be pushing mysids towards their thermal optimum and beyond. And like that thermal optimum curve, when you push them beyond that optimum, they become less able to create all of their energy for all of their different energy partitions. And so this does show that mysids are sensitive to thermal fluctuations in the winter, so potentially in the future, we might see some impacts with temperature as well. And with that, I'll hand you over. Thank you.
Thank you, Julia. I really appreciate it. I'm going to encourage everyone again to put any questions in the chat, and don't forget to introduce yourself and your affiliation and where you're joining from, so we can all kind of see who's here and where you guys are from. And next, I'm going to introduce Dr. Meredith Seeley. Meredith Evans Seeley is an assistant professor at the Virginia Institute of Marine Science, VIMS, and the Batten School of Coastal and Marine Science at William and Mary. Dr. Seeley's lab group studies the sources, fate, and effects of pollutants in aquatic ecosystems. This research is focused on organic contaminants, including plastics, plastic additives, petroleum from oil spills, tire wear particulate, and per and polyfluoroalkylate substances, PFAS. In the lab, her team enjoys developing analytical chemistry tools to better characterize the chemical and physical aspects of these contaminants. This includes understanding how contaminants evolve in the environment over time toward the goal of, oops, I realized my camera was off. I am so sorry about that, you guys. This includes understanding how the contaminants evolve in the environment over time toward the goal of describing an organism's effective exposure. Her team also uses this research to design relevant ecotoxicology studies, which often consider multiple drivers of ecosystem stress. Overall, Dr. Seeley is driven by the need to develop specific and realistic pollutant migration strategies capable of having the greatest impact for maintaining healthy coastal and marine environments. Now I'll pass it over to Dr. Seeley.
Thank you for that introduction, Kelly. I really appreciate you reading all of that. Here we go. And how lovely to follow a great presentation by Julia. I assume you can all hear and see my slides okay. Again, my name is Meredith Seeley. I'm at VIMS, and I'm really excited to be joining all of you today. To get us started and continuing on our theme about talking how microplastics impact marine species and habitat, I wanted to take a minute to recognize how truly diverse microplastics are. So we all know the standard size definition of microplastics as these plastic particles less than 5 millimeters in diameter. But if we consider a lower bound of 1 micrometer, which is like a fraction of the diameter of an average strand of hair, we're spanning over 4 orders of magnitude in size for this contaminant class. In addition, there are a variety of other parameters that are really important when we think about microplastic characteristics. We often refer to a plastic based off of its polymer type, but that is just one of the chemical complexities of plastic. There are additives that might be particular to certain polymers or shared across polymer classes, different morphologies, colors, surface textures, and chemistries that all go into making a particular microplastic what that microplastic is. And these are really important things when we think about the effects of microplastics in marine systems. So I'm going to give you 2 different research examples today really quickly to kind of illustrate how different microplastics have different types of impacts based off of some of those parameters. So in the first, we'll be talking about how microplastics impact sediment microbial communities and the biogeochemical cycles that those mediate. My collaborators in that work are BK Song, Rob Hale, and Mara Walters at VIMS. In our second example, we'll be sticking with the small stuff, but going from bacteria to viruses and talking about how microplastics exacerbate disease and might impact disease in fish systems. And so my collaborators there, I'd always want to recognize Andrew Wargo, Rob Hale, and Wolfgang Vogelbein, all at FIMS, as well as Patty Swallow at William and Mary. And I'm hoping y'all aren't hearing the noise outside my office. If someone is, please hop in and let me know. I'll do something about it. Okay, so anyways, let's think about microplastics and microbes. So one of the first connections that we made between plastic pollution in the ocean and these microbial communities was microbial biofilms forming on the surface of plastics out in the open ocean. So these are examples of some of those biofilms in these images here of scanning electron microscopy. This has really illustrated that microplastics can be a sort of microhabitat for these diverse microbial communities. But a place where microbial communities are equally important and really relevant in the Mid-Atlantic region is coastal salt marshes. So we know our coastal salt marshes are really productive areas. There's a lot of organic matter coming from the water sources as well as terrigenous sources to these coastal salt marshes. And because of the hydrodynamics in those regions, a lot of this reaches the sediments. And the sediments there have a lot of a rich microbial consortia that processes this organic matter and basically functions for nutrient cycling for the entire food web. This is kind of akin to how our gut microbiome processes the food that we eat and cycles nutrients for our body. So a really important aspect of that is, of course, nitrogen cycling. And two key parts of nitrogen cycling, which I'll talk about here, are nitrification, which is the oxidation of ammonium to nitrate, and denitrification, where we remove nitrogen, labile nitrogen from that system as nitrogen gas. An important thing that we realize when we think about these coastal salt marshes now from empirical and modeling data is that microplastics accumulate in these sediments over time as well. And so what does that mean for these bacteria that are living in these coastal salt marshes and doing these really important ecosystem services? To address this question, we did a microcosm incubation in the lab and amended coastal salt marsh sediment with 4 different types of microplastics, including polyethylene plastics, PVC, polyurethane foam, and then a biopolymer, polylactic acid. They were incubated for a period of about 2 weeks, over which we sampled for water and sediment to look at the biogeochemical cycling and the microbial communities. I'll only have a chance to jump into a little bit of the results that we found there today. Here you're looking at a principal coordinate analysis for the bacterial community structure. All you really need to know for this is that each dot is a bacterial community. The different colors are different types of plastics that were amended to the sediments in which those communities were sampled. And the further apart the dots are, the more distinctly different those communities are. So a trend to jump out you first and foremost that we have this cluster of purple dots over here. This is telling us that the bacterial communities in the sediments which had PVC added to them were the most distinctly different communities. So something is driving some differences in that microbial community for sediments amended with PVC. A second trend that's interesting that I'll highlight today is that clustering close to our non-plastic amended treated sediment here, which is our red dots, is the polylactic acid. So something similar, or our bioplastics are allowing the microbial communities to maintain a steady state similar to not having plastics in them, which is interesting. One important conclusion that we drew with a lot of other data, which I won't have time to dive into today, is that this PVC community was not only different in its structure and who's there, but also what they're doing. And so we actually saw inhibition of nitrification and denitrification in sediments where PVC was added to those. So this is a really interesting finding because this tells us that microplastics in themselves can actually alter how microbial communities structure themselves in these important coastal salt marsh sediments and how they function for biogeochemical cycling for the entire system. However, a question that you might be thinking back to based off of my early discussion on different microparticle parameters is what's driving this effect? So we had this lingering question of, is it, some physical parameters of these plastics, the surface hydrophobicity? Is it truly just the polymer itself, or is there some other type of additive chemistry? And I won't have time to dive into it today, but we suspected, and it's actually been confirmed by Mara Walters, a student here at FIMS right now, that additive chemistry is what was driving this. There's an additive in the PVC that was driving this effect. So she'll be publishing that work soon. If you're interested in it, you can reach out and I'll make sure she gets that to you as soon as it's published. But this is a really cool finding that tells us it might be something that could be present in different types of plastics, not just PVCs driving this. So now I'm going to shift gears and we'll still think small, but think about disease. So pathogenic disease and plastic pollution have been linked for several years through different studies, such as this one here, led by Julia Lamb and colleagues, where they found that disease incidence in Indo-Pacific coral reefs correlated with higher rates of plastic pollution. A lot of questions about this type of relationship remain, however. So we know that plastics have biofilms and as has been shown, sometimes those biofilms have higher degrees of pathogen load on them. It could be the case that plastics harm the host or cause some sort of damage prior to the pathogen being introduced that makes that pathogen more successful at infecting that host. Or it could be the case that pathogens and plastics just coincidentally co-occur. Like places where we have a lot of plastics, we also have a lot of disease spreading around, probably from anthropogenic impact. So a place that we realize that this might be a particularly relevant question is in fisheries. So we know that in both wild and aquaculture fisheries, one of the greatest cause of stock loss in those systems is diseases such as viruses. And something that's been established within this field for many decades is that when you introduce a pathogen such as a virus to a fishery population, the mortality or the number of fish that are going belly up as a result of that introduction is going to change based off a variety of environmental factors. And I think it's fair to say that we know that plastics are now an environmental factor, both in wild fisheries, but certainly in aquaculture settings where a lot of the gear is made of plastic that these fish are living in. So we ask a simple question, when we have this type of co-exposure, does the overall outcome stay the same? Do plastics somehow decrease the population mortality, or do we have an increase in disease severity as a result of microplastics in this system? So to test this, we used a really well-studied system called infectious hematopoietic necrosis virus, or IHNV. This virus is global. It particularly affects a variety of salmonid species, as we studied here, rainbow trout. And we used two different types of plastic, nylon microfibers and expanded polystyrene, both for their use in aquaculture settings, as well as a non-plastic kind of particle control, a ground marsh grass, Spartina, as it will be, you'll see it referred to from here on out. So just like in our sediment situation, I didn't have a lot of time to dive into data. So these are the only graphs I'm going to show you from this research. What you see here are 3 survival curves for each of our different types of exposure: nylon, polystyrene, and Spartina from left to right. If all the fish survived, then the line stays up at the top of that y-axis there over time for the duration of an 8-week exposure. And you can see that we expose fish to particles chronically over that 8-week period, with the introduction of the virus happening acutely at 4 weeks. And so what you see here is that first off the bat, all the dashed lines are staying at the top. So that means that any of our fish that were not exposed to the virus really weren't dying, regardless of what types of plastics they were exposed to. So these plastics, especially at these low doses alone, were really not affecting these fish. However, as we dive into the different particle treatments, we start to see some interesting results. So the fish that were exposed to IHNV only are in these solid black lines here, are kind of positive control. And then we have fish exposed to different varying levels of different nylon microfibers, polystyrene microplastics, and Spartina microparticles. And what you can see is as we go into these colored lines, those lines drop down, we're having lower survival overall, particularly for these nylon microfibers. At the highest concentration, which was 10 milligrams per liter, these fish had a 6.4 times higher chance of dying compared to IHNV alone. So this is significantly increasing the mortality outcome for these fish. With a variety of other parameters we tested, we also found that this was associated with higher viral body burden and some inflammation and inflammatory response, particularly around the gill tissue, that was elevated also in fish just exposed to these nylon microfibers alone. So what we suspect was happening, and have kind of continued to evolve this hypothesis with some follow-on work, is that in a typical system, when a fish host is introduced to a virus at the gill or in the gut tissue at that epithelial layer, which is illustrated here, those defensive barriers do a nice job of keeping that virus at bay. But when we introduce particles such as microplastics, that might cause some sort of inflammation and tissue damage that allows greater host colonization, reduced immune defenses, and that might be more enhanced for these longer, more rigid nylon microfibers. So, as we think about what this means in respect to the first conversation about sediment microbial communities, we really have two different stories here. One where the chemical additives are driving an effect, and one where the plastic shape is driving the effect. And although we tend to talk about plastics based off their polymer type, just like I did throughout the entire conversation, we really see that polymer type is probably not what's actually driving that effect here. So, when we think about microplastics and their impacts in these different systems, when we try and match up these lab exposure studies to what's in the environment, it's really important that across both of these types of experiments and surveys, we're testing a variety of different particle parameters, including these size, shape, physical aspects. When we do that, we can start to look actually at informing risk for different systems. So what we're seeing as really impactful for certain different types of microplastics, for example. We might also be able to reduce the number of measurands that we have to capture for plastics in the environment. If fibers are the issue, maybe we just count numbers of fibers, for example. So I think this is a really positive outlook for the future of microplastic mitigation, regulation, and monitoring. And with that, I want to say both of these papers, I breezed through them, but they're both published, so you can follow those QR codes here. I, of course, want to thank our funders. Here are our collaborators again. And their work wasn't mentioned in this, but my lab at VIMS is the Coastal and Marine Pollution Lab, and my students Robin Dudrick and Liam Green are doing great work with sediments, with seagrasses, tire wear particles, and more. So if you're interested in what we're up to now, please reach out. And I will stop talking now.
Thank you, Meredith. That was great. Okay, we have one more speaker. Our third presenter is Dr. Hayden Betcher, who is another UD grad, a fellow Blue Hen. Dr. Hayden Betcher is a marine scientist whose doctoral research focused on the distribution and physiological impacts of microplastic debris in the Delaware Bay ecosystem. Specifically, his work addressed three key questions. Do microplastics and planktonic crab larvae overlap in space and time in the marine environments? Two, are crab larvae ingesting microplastics in their natural habitat? And if so, to what extent? And lastly, in controlled laboratory settings, how does microplastic exposure affect the growth and development of crab larvae? And how do these effects compare to those caused by similarly sized natural particles, such as sand grains? Dr. Betcher earned his bachelor's degree in zoology and conservation biology from the University of Wisconsin in Madison and his PhD in marine sciences from the University of Delaware. He is currently a teaching postdoctoral fellow at Providence College. In addition to his research, he's deeply committed to education and is passionate about training the next gen of marine scientists. He's actively building a career dedicated to this goal. And with that, Hayden, I'll pass the torch to you.
Thank you so much, Kelly. I will pull my presentation up here. Okay. Unless I hear otherwise, I'm going to assume everybody can hear and see me okay. Thank you so much for the introduction. My name is Dr. Hayden Betcher. Today I'm going to be talking about that third kind of research goal that Kelly described a few seconds ago, which is zooming in on, like, let's take this to the lab in a very hyper-controlled setting and see what effects we can tease out of how microplastics and microfibers can have an impact on the development and physiology of crab larvae. And so initially, when I set out to do this research, I really wanted to target blue crab larvae, just because they're really important both economically and environmentally. But when I tried doing those experiments in the lab setting, the amount of mortality in the larvae was just too high to get really good data out of. And so we ended up switching to a surrogate species, which is the mud crab Panopeus herbstii. And so today I'm going to be talking about the data and results we collected from those experiments. And so there was one big kind of overarching question here that we're trying to answer, which is, what are the effects of microplastic exposure on these larvae? And so we came at it from two different angles. The first angle was using developmental experiments. And so we let these larvae rear and develop in the lab setting under a couple different treatments of microplastic exposure. And so the microplastics we used were polyethylene terephthalate microfibers. So this is PET. And as Meredith was saying, the size and shape and formulation really has a different impact. And so we were using 50 micron fibers here. And during these developmental experiments, we introduced a few different treatment levels of microplastic exposure. So we had a 0, which is our control, that's 0 microplastics per milliliter. And then we started increasing exponentially from there up to 0.1 pieces per milliliter, 1, and all the way up to 10 microplastics per milliliter. And during these developmental experiments, we tracked a couple different endpoints. The first one was survival. And then we also looked at development time. So how long did these larvae stay in their specific developmental stages? And then the third was body size. So kind of a proxy for the physiology of what's going on. And then to follow up these developmental experiments, we also did a grazing and respiration experiment. And so here is where we started to include these natural sand microparticles as a positive control for microplastics as well. And so we had microplastic treatments, control treatments, and also a natural particle treatment, all at our high end of exposure, so 10 pieces or particles per milliliter. And during these experiments, we measured the grazing and respiration rates. And so we're going to get into that later. First, we're going to talk about the developmental experiments. And so for those of you who don't know too much about crabs, crabs undergo this really interesting metamorphosis as they work their way through development. And so they all start as what we call Z1 larvae, and they molt in very distinct steps from Z1 all the way up to Z4, and they kind of get bigger and change their morphology as they go. Then they morph into megalopae and finally into what you would recognize as an adult crab. And so what we wanted to do was expose the larvae across their development here all the way through megalopae. And so during these developmental experiments, we ran 3 different experiments, kind of modulating microplastic exposure from each of the 3 experiments. And so the first one was a full exposure experiment. Basically, this just means you had microplastics present in the water the entire time from Z1 all the way through megalopae. The second one was an early exposure treatment. So we had exposure all the way through the start of the Z3 stage, and then we took all of the plastic away and let them continue to develop into megalopae. And then experiment 3, which was the late exposure, was the exact opposite of that. We had them in control conditions at the start and then moved them into microplastic exposure afterwards. And so here's a brief look at the experimental setup for these developmental experiments. As I said before, we had 4 different treatment levels increasing from 0 to 10 microplastics per milliliter. All of the larvae were sealed in 1-liter glass jars, and they were rotated on this plankton wheel here to ensure that the microplastics were constantly being distributed evenly in the jars. We worked really hard to make sure that there weren't any air bubbles in the jars because the plastics like to kind of congregate around air pockets and things like that. And we did water changes and data collection every other day throughout the 30-some day developmental period for these larvae. And in total, using the 3 jars for each treatment, we tested 90 individuals for each treatment. And this whole setup was repeated 3 times for the full exposure, early, and late exposure experiments. All right, so let's get into the results here. First, we're going to be looking at the impacts on survival, which were inconsistent, to be perfectly honest. Here we have Kaplan-Meier survival curves. This is very similar to what Meredith showed before, where you have your overall survival probability on the y-axis. And so if you stayed alive, you would stay at this 100% probability of survival mark. But because we're working with these planktonic larvae, mortality is really high in general. And so you're going to see, even in the control, a lot of die-off all the way down to about a 50% survival rate. And that's pretty normal. And we don't actually see any major differences in survival with microplastic treatments increasing. And so in the gray here, we've got our control. And then as you get into the darker red, so from the pink to the darker red, all the way to the very, very dark red, you're increasing in concentration. And you might say, oh, maybe we can tease away a little difference between the two lowest concentrations and the two highest. But what I'm not showing here is the standard deviation. When you start to add that in, it looks a little bit like a jumbled mess here as all of that variability starts to stack on itself. And so we didn't actually find any significant differences in survival during this full exposure experiment. So when we exposed them from day 0 all the way to day 25. Now, next, I'd like to take you through the early exposure and the late exposure data for the survival results. Here, again, you could tease away some actual significant differences. So for the early exposure, we had microplastics present during this gray shaded area. And then around day 15, we took the microplastics away. And what you see is a significant difference between the 0 and 0.1 pieces per mil treatments, so our two low treatments, compared to our two highest treatments. And so you might be thinking, okay, maybe there's something there, maybe there's some impacts on survival. And so when you ran that again, but this time with late exposure, uh, here we've got a control condition for all of the treatments, so no microplastics present during the initial 15 days. And we still see significant differences in survival even before microplastics are even introduced to the system. And so that really threw a bit of a wrench into interpreting all of this data, where you see significant differences occur, but they also occur when there's no microplastics at all. And so there's just overall really highly variable survival in these planktonic larvae, which is to be expected. So next we're going to talk about a decrease in body length. And so here we measured the body length of the larvae as they went through these different treatments. And what we found is that high levels of microplastic exposure resulted in a decrease in body length for these larvae. And so on the y-axis here, we have body length getting larger as you go up on the y-axis. And then we've got the 4 different developmental stages that we tracked body length for. And so for the full exposure experiment, you start to see in these dark pink and dark red high microplastic treatments that you get a repression of body length, a decrease, as the amount of plastic in the system starts to go up. And that remained consistent in the early exposure experiment and also in the late exposure experiment, where you don't see any difference until plastic exposure starts to be included into the experiment. Now, I do want to direct your attention here to the early exposure experiment because we found something a little interesting here in that you see an initial decrease in body length with microplastic exposure. But after microplastics were taken out of the system for all of the treatments, we saw evidence of recovery for these larvae. And so that suggests that they're able to kind of divert their energy usage more into catching up in size compared to when they had microplastics in the system, which was really interesting. Next, we're going to talk about developmental delays. And so here it's basically how long did the larvae spend at the Z1 stage before molting into Z2 or before molting into Z3, etc. And so here we're tracking how long they're stuck at each of their developmental stages. And so again, we see a pretty consistent delay with increasing exposure of microplastic. And so here we've got essentially the amount of time it takes to get through that stage on the y-axis, increasing in time. And so you see for each of the 3 or 4 stages that we track this for, the more microplastic in the system, the longer they actually stayed at that stage compared to the control. And this remained consistent for the full exposure and late exposure experiments. And then again, in the early exposure experiment, we saw evidence of recovery after the microplastic exposure actually ended. So the amount of time they spent in that last stage actually matched what the others were after the microplastics were taken away. So next, I just want to touch on the grazing and respiration experiments. The previous ones were done in groups of 30 per bottle. Here we actually looked at individual grazing and respiration, which was really great to get that really fine granular look at the data. And so here we actually exposed individual zoea to one of three different treatments, either 10 microplastics per milliliter, 10 sand particles per milliliter, or a control that had no particles whatsoever. And in each treatment, we gave the individual zoea 10 prey items. So 10 artemia that they could eat ad libitum. And we measured how many food they ate in a 24-hour period, and also the respiration rate of oxygen consumed per hour. And so the respiration rate is just a way to get at their physiology or their metabolism, what is actually going on, how much energy are they spending. And so here we did 20 individuals per treatment, and we got some pretty interesting data out of this. And so to start, we looked at the grazing rate. So this is how much food are the larvae actually eating per 24 hours. And if you look here on the y-axis, we have exactly that, the number of food or prey items eaten per day. For the control group with no particles whatsoever, they're eating about 5 of the 10 available prey items per day. If we move on to the sand or the natural particles, the natural microparticles, we saw about a 21% decrease in food intake, so down to 3.7 as an average. And then moving on to the microplastics at the same concentration and the same size as the sand particles, we saw that drop down again to a 43% decrease. Excuse me, a 43% decrease in food intake, which is nearly half as much as the control, which is a pretty impressive decrease. And so overall, we saw that both the natural sand microparticles and the plastics decreased the amount of food that the larvae ate, but the microplastics had a larger and more significant effect. And this pattern continued for respiration rates as well, but in elevating respiration rates. And so here we have our baseline or control respiration rate of 0.26 micrograms of oxygen per hour. This is kind of like our baseline. It's pretty reasonable. But when they're exposed to natural microparticles, that increases by about 48%. That's a big increase in respiration rate, a big increase in how much oxygen you're sucking down and energy you're expending. And then again, it bumps up when you're exposed to microplastics, going up to a 55% increase in respiration rate. And so again, we see the similar effect of natural microparticles have an impact, but not as high as the plastic particles. And so we got a few key takeaways here, which is that PET, microplastic fibers, impacted the body size and development time of mud crab larvae over the course of their development. As we looked at the body size decrease as plastic concentrations went up, it took larvae longer to reach the next stage when they were exposed to plastics. But they showed a propensity for recovery once microplastics were actually removed from the system. And crab larvae that were exposed to these high microplastic concentrations were actually able to acquire less food and were also spending more energy each day or respiring more. And we saw that natural microparticles like sand, so any particulate in the water may have similar effects, but they're less extreme. And so having these microplastic-induced developmental delays or body size suppressions forces the larvae to spend more time in the plankton at smaller sizes, which, if you can think about it, I mean, the plankton is a terrible place to be if you're a crab larvae because everything wants to eat you. And so the more time you spend there, the larger the chance of mortality, basically. And so you're going to end up reducing the number of larvae that can successfully develop into adult crabs. And as you all are well aware, having less larvae survive to adulthood could have significant impacts on fisheries and ecological services that depend on healthy adult crab populations. And so thank you so much for having me. I'm excited to have this kind of talk weaving all of myself and Meredith and Julia's research together because I think it all kind of pings off each other really well. And so I'm excited for the discussion portion, but thank you so much for having me. And I'm happy to answer any questions. Thank you.
Thank you, Hayden, and thank you to Julia and Meredith as well. I agree. I think all these top talks today really complemented each other well and learned a lot. So now we're going to move into the facilitated dialogue section of this. We have about 20-25 minutes. So not that you guys haven't spoke enough, but we're going to start it off with a question to all of you to see if you actually have questions for your fellow presenters. Oh, hi, Kat. I think the cat has a question. If you have any questions as you were listening to your fellow presenters that came up and that you wanted to ask to kick off our dialogue this afternoon. So don't be shy, just hop on in. We don't need to do
Right.
the whole raised hand things.
I'm gonna jump in with Hayden because it's fresh on my mind. So these polyester particle or fibers, right? I'm just so curious. I know you didn't test like different lengths and things, so you can only test so much, but your gut feeling from doing this research, do you suspect that longer fibers would have a similar impact, or is there some of this that's driven with like some parameter that only the small 50 micrometer, I think you said was the length, could really get in there?
So at this scale, we were really working hard to mimic the size of natural prey
Yeah.
particles. And so that is why the 50 micron size was chosen. Um, as you said, like size is really variable. It's really important. It probably alters the effect. I would actually imagine that having larger sizes of microplastics would decrease the effect.
Yeah.
I think a lot of what is going on with the effects we see in reduced grazing, particularly, is that they're handling and trying to eat these particles, and they're just spending time and energy wasting on trying to eat something that is, for all intents and purposes, to them, a potential prey item.
Yeah.
And I think if you increase the size, you may end up seeing less of an effect because of that.
Yeah.
Um, Something that I didn't have a chance to talk about in the presentation in and of itself is that we didn't actually see a ton of ingestion of these particles in the lab. We tested that a lot with different sizes of plastic. It happened. You could get it to work, but it didn't happen at a rate that would explain all of the decrease in grazing. And so I think it's more of a handling or like energy expenditure issue.
That makes sense. Thank
Yeah.
you.
Any other questions for your fellow panelists? Otherwise, I can keep moving on. We're good? Okay, next question. This is a one that I'm sure we've all been asked many times, but what research gaps still remain in this field of study that you guys focused on today? And is there anything you wish, if you had unlimited resources that you could go back and research specific to this Mid-Atlantic region?
If I had unlimited resources, I would want such great monitoring data with really fine scale resolution on all of the parameters that we're interested in. Because I think that's really a struggle right now is our throughput and what we can measure in the field versus what we can study effects wise in the lab.
I hear that.
Yeah, I know you do.
Anyone else have any ideas on some research gaps that we should be prioritizing and maybe something you wish you could have studied specifically in addition to the research you presented on today?
I'll jump in really briefly and then let Julia go as well. I really think that the work that Meredith and Julia presented, which is like this idea of going into the field and actually doing experiments in the field or getting these experiments done not just in the lab setting, but in like a field-based microcosm kind of an experiment. They're really difficult, but they're really important to do. And to piggyback off of that, the natural microparticle stuff that both Meredith and I incorporated in, and people are incorporating this more and more now. But it wasn't done maybe 4 or 5 years ago. I think is really important because you could, even in the results that I found, like there was actually an effect of just sand being in the system. And so often our control experiments are just clean, filtered water. But in the natural environment, that's not what these animals are dealing with. And so I think shifting our baseline relative to perfectly pure, clean water, I think is really important as well.
So with the field assessments and, advocating for that as needed, what is the reasoning why, I guess, more field assessments aren't being done? Is it timing? Is it resources and funding? Is it access? Is it all of those things?
Yeah, I think I can jump in there. I think it's almost all of those things. So, doing that field work was really tough. It was in the middle of the night. It was constantly going out, driving hours out of my way, and that's tough. But I was a student, you just do it. Um, but funding is really hard. Sometimes the misons aren't even there. Your species might be gone for some reason, so it's all kind of these things that fluctuate. And then you bring them into the lab, and now you also are introducing all these lab effects. So it's really hard to tease out. So I tried my best in my project, obviously, to tease out these like field versus laboratory anomalies, but it's really hard to say, yep, plastic is what's driving this. Yep, this is what's happening. So with our current data sets, so it's really hard to be like, I didn't stress them on my drive back home, and I probably did. But to answer some of the questions in the thing, I did acclimate them for 24 hours so they weren't just thrown right into a respiration experiment. But yeah, so it is tough to study in the field. But I do, to piggyback off of everyone, I think that is the most important thing because currently we aren't seeing the concentrations that laboratory experiments are showing effects of. So usually laboratory experiments are showing like 10 to orders of magnitude higher of what's in the field currently. And that's not always the case. So the field is a point, right? You do a microplastic tow and you get at that time, at that exact moment when the tides were exactly right, when the current was exactly right, you're only getting a snapshot of what the plastic is like as a whole. So it is kind of like you win some, you lose some, but there is some
Yeah.
part.
It sounds like a double-edged sword the way you're describing it, where in the lab you can create these perfectly I don't want to say unrealistic, but these perfectly clean
Right.
scenarios where you can isolate everything. But in the field, you're seeing the more natural loadings and more natural variabilities, but then you can't really isolate the drivers of why those variabilities are happening. So it sounds like we need more coupled field and lab studies to
Yeah.
really kind of isolate that. And I saw Christine had her hand raised, and I think she put in the chat a specific question to the dialogue we're in, which is a question for Meredith on what specific parameters would you want to be able to monitor. And Christine, if you want to come off mute, if you have the ability, if you need to add on to that, feel free.
Thanks. That's yeah, that's the whole question.
I did it. I read it wrong.
Yeah. Thanks, Kelly.
Nailed it. Great question. Like all of it, right? So following up on the recent conversation about just capturing a snapshot in time, I think part of the monitoring data would have that kind of temporal trend to it, and particularly allow us to hone in on some hot spots. Because while I agree a lot of time in lab studies are using concentrations that are higher than lots of environments, they're certainly not irrelevant to certain places where you might have like a microfiber outflow on the back of a production facility or something. So it's really important to capture all of those kind of fluctuations through time. I would definitely want to see polymer type, size, form, and fiber fragment bead, as well as some sort of future adaptability for additive scoping as well. So particularly with our larger plastics, I think we're already there with like PyGCMS and other tools that allow us to really get into what types of additives might be present in particular plastics. But we don't do that so often. So I would like to see more of that.
Thank you, Meredith.
Yeah.
All right. Oh, Avalon raised your hand. Go ahead, Avalon.
Hi, yeah, and thanks to everybody. These were such fantastic presentations. You all are doing wonderful work. And I was wondering, I think maybe in every presentation you sort of alluded to this a little bit, but Marco is it's a partnership between people that make decisions for coastal management. And I'm just wondering what your thoughts are, and this is for any or all of the panelists, what your thoughts are on what else needs to be done with this research in order to get to recommendations for policy and management. And what's the timeline on that? Because it also seems to me that, research on microplastics is somewhat new. And so do you have a sense of the scale of time? Thank you.
Who wants to take that one first? Yikes.
It's a good question. I mean, I can take a stab at it because I think it might not be so much a question of time, but a question of funding. So I think we're there as far as like getting samples done, throughput. Like I would love to monitor more. We're ready for it, right? What we don't have is enough funding to support the people who have to do that work. So it's not an easy assay where we can just measure things and it's low bench time. It's a lot of time to get there. So it's hard for me to predict what would happen with funding to think of that in the context of time, but I think that might be one of the big barriers.
Yeah, just to piggyback off of Meredith's point, this kind of work is really bench work intensive a lot of the time. Like I can at least speak for Julie and I, who sat next to each other for the better part of 4 years, picking plastic particles out of filters and weighing out just the right amount of plastic so that all your treatments are exactly the same. And it takes time to do that work well. And the more people who are working on it at the same time will really expedite things. And so I think that's where the funding comes in. Um, I would actually prefer to, or take the chance at least to flip the question on you, Avalon, which is like from someone who's like having these conversations with decision makers, what are the benchmarks that they're looking to meet in order to enact policy change? Because I think it needs to be like we need to be working towards the correct benchmarks so that like all of the data that's being produced actually has like a foothold for whoever's actually making the decisions, that it's not just kind of going into the nether and not being used to actually enact those decisions.
Yeah, that's a great point. And I think that this is, when we think about benchmarks, to me, that's really the nexus of where science and policy meet, right? You all are the ones who are informing what those benchmarks should be. And then the states and other decision makers are determining the capacity and funding for being able to make sure that their policies are helping meet those benchmarks, and configuring out what a monitoring protocol would look like at scale. Because obviously at scale, you're not going to be able to have, all of the states having hundreds of people counting plastic, having some mechanism for easy monitoring, I think is probably the first step. But I'm not a decision maker myself. We do have some folks on the line today. I will encourage people from the states to speak up, or others on the line, but that's my gut reaction there.
Yeah, I mean, hearing all this, it sounds like there might be some opportunities for more of a regional collaboration or opportunities to pool those resources when it comes to monitoring, or when it comes to, I guess, collecting the data that we need to make measurable change, whether it be policy or whatever. So do you see any opportunities, or are you aware of any opportunities that people should get more involved in, that a place like Marco could help spread the word or something about getting these kind of collaborative opportunities off the ground?
You teed me up nicely, Kelly. We're having a summit at the end of this year. It's a regional marine debris summit, and we're in the process now of developing the agenda for that. Concurrently, Marco co-hosts, along with Maricus, the Mid-Atlantic Coastal Acidification Network, which is not about microplastics, but it is about regional scaled water quality monitoring, and thinking about how to institute acidification monitoring into existing state water quality monitoring protocols. And we often through MACAN will have discussions about regional monitoring needs. We've done regional hotspots analyses and gaps analyses and all of that. And I kind of see some similarities in the approach, some conceptual similarities. And I wonder, Will, I'm sort of looking at you, if we could think about the potential to have, a monitoring discussion during the summit that could, extend this a little bit and bring all of you experts to the table to talk about it with decision makers at the same time.
Yeah, I think that would be a great opportunity. And given all the work that I know that Christine is working on, kind of compiling a lot of the monitoring that's going on, a lot of what the plastic pollution action team in the Bay program is working on, it's a good time to be having those conversations. I was writing this into the chat just to kind of respond to the question of what do decision makers want to know, and so maybe as kind of a preemptive thought for that conversation we can have at the summit in the fall. I think one of the problems with microplastics is what can you do about it? And as decision makers, the big thing we want to know is if there's something to do about it, information that leads towards those actions is what we want to know. So when Meredith was talking about specific additives that may be more troubling, is there something that we can look at that and say, we're going to try and limit these types of plastics entering our environment, plants that are producing it need to have more specified waste treatment or something like that on their systems. Those kinds of things where otherwise, if it's just microfibers are bad, I think a lot of us, as we talked about in our first webinar, they're really hard to control because they're everywhere and we're all wearing them. And so how do we get towards those control mechanisms? It helps to know what's possible to then design the policy around it. But anyway, I digress.
I mean, I think that's an interesting point because that's the question we always ask is how much is too much? And then so what? Like what happens next, right? So I think the opportunity to work with Marco and attend that summit seems like a great place to look at monitoring and then look at mechanisms for control. So I'm looking forward to hearing more about that. I have a couple of other, I think, specific questions for some of you. I know we only have like 5 minutes left, and I really want to be respectful of everyone's time. Julia, there was a couple questions that came in around, have you done any testing during the warmer months? And then what time of day did you test? And then I actually had a question with how does water temperature, potentially impact degradation rates of plastic? So if we're seeing a warmer trend, are we going to see longer or shorter residence times in plastics or certain types of plastics that may exist longer? They're not as impacted by warmer temperatures. So anything you could speak to that would be really great.
Yeah, totally. So I just popped an answer to the question in the chat just to have a longer answer there. But I did test them in the summertime, only at that high plastic location, and we found that the rates did double, and we expect that. But at that high temperature, so we kind of pushed them on a curve similar to what I did in the winter. And at 30 degrees, they doubled again, just like that 10 degree showed. So we're seeing that stress at really high temperatures these animals are not adapted to at that time. So we are expecting a double in rate over the summer. That's pretty normal physiology. However, pushing them past their limit, they do react to that. When I tested them, I would collect them at the evening. They're usually at the surface at that time, so I would collect them at the evening and then test them the morning after. So I would put them in their temperatures overnight, acutely adjusting them to the new temperature treatment, and then tested them the following day over 24 hours to answer those. And then to answer your question on plastic degradation, that's tough to say. Residence times are always tricky when it comes to plastic, especially microplastic and then especially nanoplastic, because what it does is just breaks down into smaller particles, right? Or leach chemicals. And that's really tough to test if you don't have the virgin material that it came from. So I can't speak to really how fast or slow it might uh, like digest out. But we do have some really cool modeling work to show how tides and spring versus neap tide currents and circulation might cause aggregations of plastic and where they might aggregate in the water column. And that can change based on winter and summer. Summer being a more stratified system and winter being a more well-mixed system, and in the Delaware Bay especially. And so that can aggregate plastic and also zooplankton that can't swim against that current. And so it kind of causes these like microcosms almost in the ocean or in the estuary that kind of push plastic and organisms together. And those are where I would like to keep pushing my like pen in and see what's going on there. Because they can get like what, like 5 times the average plastic concentration in those regions. So it's pretty interesting.
Yeah, wow. Thank you so much. We only have 1 minute left. So I really want to be respectful of everyone's time, especially our presenters. So really quickly, do any of our presenters have anything they want to share or plug on some projects that they're working on right now in this space that we could all keep an eye out on? I know Meredith mentioned one of her students is going to be putting a paper out soon. So I just want to pass it around to the presenters and see if you guys have anything you want to share.
I guess for me, that paper of accumulating plastic should be coming out later this year. And so if you want to keep an eye out for Fontana et al., that should hopefully be out later this year. Cross your fingers.
Awesome. Hayden, anything you want to share?
Yeah, I also have 2 papers that should be coming out within the end of the year as well, if all goes well as well. One of them also looking at this aggregating factor of plastics and crab larvae, as well as the in situ ingestion of microplastics by crab larvae, which we've found surprisingly a lot, like more than what we could get them to do in the lab. So that was interesting. And then the other one is the work that I presented today. So that'll also be coming out shortly. Other than that, the last year and a half, I've taken a little bit of a break from microplastic research. After the 5 years, I
break.
needed a little bit of a break. And I went on and did some neurobio research and kind of learned how to do EM microscopy and confocal and all this, more of like a techniques focused postdoc, as well as some teaching. But this conversation has been great. I think I've gotten bit by the bug again, and so I'm excited to keep the
We're gonna
conversation
suck you back
rolling.
in.
Yeah, sometimes you just need to step away for a minute, and then you find that motivation again. And so yeah, I'm interested to keep this conversation rolling, and I'd love to connect with anyone in the chat or even other presenters today. I mean, this has been a lot of fun. So thank you again.
Anything you want to share, Meredith?
I think I already covered it. So,
Awesome.
yeah.
All right. Well, I'm going to give a virtual round of applause to our great panelists today. Thank you so much. This webinar is going to be available online. I will put the link in the chat. It'll be on the Marcos Marine Debris Work Group webpage. So keep an eye out for that. And keep an eye out for a future invitation to the Marco in person or the Marco upcoming fall retreat. And then I think the big takeaway is we need more field assessments. So let's talk in the future. And I appreciate everyone's time and thank you so much. Have a good one.
Thanks, Kelly. Thanks, everyone.
Bye, everyone.
Thank you all.